So the next topic under nucleic acids and proteins is the process of transcription. Transcription is one of the processes involved in protein synthesis. So prior to us explaining the process of transcription, since this is being learned by HL students, we have to review them on certain starting points. So what are those starting points? Because I believe this transcription has been already explained partially in the chemistry of life, wherein simple terms has been used and not all details are being discussed in that area. So we have to review on certain starting points, such as their knowledge about the DNA structure and their knowledge about a gene. Okay, so in this case, since that we have to review our students on their knowledge about DNA structure and gene, we are now going to relate the significance of the DNA and the gene. So the DNA consists of several genes that will code for a specific protein. So since the genes basically dictate the order at which specific amino acids that are going to be assembled together, then we are able to achieve the process of protein synthesis. So we should be able to make an overview of what protein synthesis is. So we should be able to tell our students that we have to, it's important to know that the gene dictates, or basically the DNA dictates the information or the sequences of bases at which specific amino acids that are going to be assembled together into a protein. Okay, so the third point that is needed to be discussed under the process of transcription is the comparison between sense strand and antisense strand. Okay, so for that matter, we need a diagram of the diagram of the DNA, and then we need to, for example, we have this. For example, okay, so for example, we have our DNA strand here. Okay, so in the process of protein synthesis, or in the process of transcription, both DNA is actually both DNA strands shall be labeled as. Actually, one of the strands in the DNA will be labeled as the sense strand and the antisense strand. Okay, so now we need to differentiate what sense strand and antisense strand. So the difference of transcription and DNA replication is that in DNA replication, as a review, both DNA strands are going to be used as a template for the production or for the formation of the new DNA strand. And here, in the process of transcription, one of the strands is going to be used. Okay? So the DNA strand that is going to be used as the template onto which we are able to attach the nitrogenous bases on the template strand is what we call as the antisense strand. Okay, so this will be our template. And then okay, so the partner strand is what you call the sense strand or the coding strand. Okay, we should also lay down the significance of this two strands. So if we, it is also important to make a matrix on the differences of these two structures. So in the case of antisense strand and sense strand, okay, so this is the one that is going to be the template, wherein we are going to attach the nitrogenous bases on the antisense strand, whereas the sense strand will be the coding strand or the partner strand. Okay, so the partner strand or the sense strand contains the promoter region. So the promoter region, this is a region in the DNA wherein it is going to be recognized by the RNA polymerase, the enzyme that is supposed to be used for the process of transcription, so that this is where the RNA polymerase is going to attach itself. Okay, so in the case of antisense strand, okay, so it's going to be, it has the same base sequence as the tRNA. Okay, for that matter, we have to include as well on the starting point their knowledge about the types of RNA. Okay, or RNA structures such as the mRNA, tRNA, and the ribosomes. So, in the case of same base sequence as the tRNA, we have to take note as well that the T, okay, the nitrogenous base thymine on the DNA strand will be replaced with U in the tRNA. Okay. So, same way, in the sense strand, this contains the same base sequence as the mRNA. Okay. Again, T will be replaced by U. So here we can say that for us to be able to check whether the mRNA strand that has been formed is correct. Okay, so for example, we have the mRNA here. The mRNA and the sense strand share the same sequences of bases. Okay, for us to, for that matter, for us to be able to check whether correct nitrogenous bases has been attached on the template strand. Okay, so there. So in the case of the comparison between sense and antisense strand, we have to remember the diagram of the DNA and labeling both strands. Okay, whether it's going to be the sense or antisense strand. And we have to provide a matrix as well, okay, to show the comparison between the two structures. Okay, so the next part that is needed to be learned by most students, learned by HS students in this process is, of course, the process of transcription itself. So first, we have to make an overview of what transcription is. So basically, the goal of this process is to form a complementary strand of the DNA, which we will call as the mRNA, okay, or the complementary copy of the DNA, which is the mRNA. Okay, so the mRNA molecule is going to be formed with the help of an enzyme called the RNA polymerase. So we have to include what should be produced at the end of the process. Okay, so we have to include the product, which is the mRNA, and the enzymes that are involved in this process, such as the RNA polymerase. Okay, so it is also best if we are able to provide a diagram, okay, for the process of transcription, so that the students will be able to visualize on how the process is occurring. So providing a diagram or providing a video will be much better, because in that way, okay, the student will have this image of how the process is occurring. Okay. So lastly, okay, we have to include as well the differences between the process of transcription in a prokaryotic DNA and a eukaryotic DNA. Okay, so in the case of transcription process in eukaryotic cells, okay, there's the presence of the promoter region, which is going to be recognized by the enzyme RNA polymerase. Okay, so actually there are several promoter regions in the length of the eukaryotic DNA, whereas for the prokaryotic DNA, there is only one promoter region. So another thing that is different between the transcription process in prokaryotic cells and eukaryotic cells is the presence of the regulator proteins in the eukaryotic DNA. So this regulator proteins needs to be activated first for it to be able to continue the process, or for it to be able to allow the process of transcription to occur. Okay, so prokaryotes and eukaryotes. Okay, so under this area, okay, we need to include as well the presence of promoter region, regulator protein, or regulatory protein, and the process of post-transcriptional modification. So this process of post-transcriptional modification will be discussed later on. So these are the things that is needed to be discussed under the process of transcription.